much, Carl. It's really fascinating and unbelievable. Now you can understand why I say that he is the promising scientist in our health system. And we will move now to the last lecture in this session, the lecture entitled The Molecular Medicine Revolution from Vaccine to Genome Editing, and it will be given by Professor Dan Pe'er. Professor Dan Pe'er is the Vice President for Research and Development at Tel Aviv University, where he heads the Laboratory of Precision Nanomedicine, and from 2016 to 2020, he was the chair of TAU Cancer Biology Research Center, the largest of its kind in Israel, and since 2017, he has served as a founding and managing director of the Spark Tel Aviv Center and for Translation Medicine. So the podium is yours.
Okay, so first I want to thank the Samuelis. I think it's an amazing, really opportunity to be here, present what my lab is doing and not what the university is doing. So, and also I want to thank you for your generosity. I think this is amazing for both the engineering, the medicine, and science in general. So we are living in a revolution. Yes, my friends, we are living in a real revolution. Because by the end of 2021, about 2.5 billion people will receive a new therapeutic modality, an mRNA entrapped in lipid nanoparticles as a vaccine. Now, I can tell you that exactly 10 years ago, I submitted a grant to the NIH with the title mRNA entrapped in lipid nanoparticle, potential for new vaccines and therapeutics. And it was not reviewed even. And when I appealed, the grant officer was very kind to me, and he says, I should actually write it down because it's nice. He said, this is a visionary grant that we do not foresee happening. Therefore, the NIH will not fund this. Actually, I sent it to Francis Collin about half a year ago. And he responded very nicely, said, well, you could not imagine, something like this. So I think Broadcom, and actually what Professor Samueli has done, is actually very similar. So he was also living in a revolution and made the revolution. So thank you. Okay, so our lab is studying how to manipulate cells in order to generate novel therapeutic approach to treat mainly leukocytes-implicated diseases. And this is a huge field that includes blood cancer, inflammation, viral infection. I should actually add corona as well. But we are living in the field of RNA for the past almost 20 years. And therefore, we have to, of course, copy from somebody smarter. Okay? And so many things happen over the past years, really, and we learn lots of tricks from nature, from the DNA and from the RNA as well. So my lab is divided into two major groups, okay? I would say the engineers and the biologists. The engineers that do everything from synthesis of lipids, I'm going to spare some time on this, engineer proteins, and generate antibodies. And the other part, we're doing the biology, try to understand and discover new potential drug targets. And of course, in an ideal world, you want to combine both of them. So you want to identify the target, and you want to deliver it specifically to the right cell. Oops, yes, this is it. Okay, so since I want to educate you a little bit about what you have gotten, and if not, you're going to get, I hope you all gotten this. So basically, we're talking about lipid nanoparticles that are almost all natural, except for about 50%, which is completely synthetic, which is an ionizable pH-sensitive lipid. So since we also discovered the mechanism by which cationic lipid can do harm things to the immune system back in 2010, we drove the field into pH-sensitive one, okay? And Srinivas Ramishetty from my lab took this molecule that was approved for clinical use in the FDA in 2018 with an island product, which was the first siRNA to silence a gene in a rare genetic disease, and basically did what we call in Hebrew reverse engineering. Okay? So he took the molecule and basically changed the molecule, the head groups, the linkers, and the hydrophobic tails, and came up with a lot of other new lipids. And so we started working on this a few years before it was approved for the FDA, and we use microfluidics in this, and anyhow, what happened is we started to collaborate with a small German company, about 30 people, called BioNTech, back in 2014. This company is behind the Pfizer vaccine, and one of the experiments that we have done, it's a lot of data, but basically one of the lipids you see here, EA2, is a very similar one that there is in the formulation. I'm going to show you how it works. This is an experiment that was done in non-human primate, basically to look at safety of liver enzyme back in 2018. And this is how the particle looks. I don't know if you can see, but they're a very nice round-shaped particle, and this is what we get. So inside you have an mRNA, and that could change the world, basically. So what we have here, and what I've talked about, is actually a first part. So we can make those particles, we can scale it up. It's about, the most one is about 50 liters. You can do it in one day. So just imagine the entire population of Israel is about 12 days of work. Okay? So you can get about 10 million vaccine in 10 days or 12 days of work. The problem starts when you want to go to the next level, the next generation. And for the next generation, we want to create science fiction. We want to give an address to those nanoparticles, to make sure that they will target specific cells, but not the other cells. And we want to deliver very important payloads, mRNAs, siRNAs, CRISPR compounds, etc. So we were dealing with this because this is not the reality. Well, what you can see here is actually not a reality. This is my imagination. Okay? So it never looks like this. And chemistry has a lot of obligations and challenges. But at the end, we decide to move from chemical conjugation, when you want to conjugate an antibody to the surface of lipid nanoparticle, and do it with biology. So chemical conjugation has limitations. Some of them are very toxic. Antibodies, for example, that you want to conjugate to give this address to a specific receptor is also a very challenging issue because it's never in the right orientation. So you cannot really orient the antibody on the surface of lipid nanoparticle and control it. So it's an engineering problem. And how we deal with engineering problem? With biology. So we went back to biology and created what we call a universal linker. Just imagine that you are now going away to different countries with one adapter, and it will work without changing the adapter. So we create a lipoprotein, basically a small adapter, kind of an antibody, that have one angle goes to the lipid, the other one goes outside and can bind any antibody from the same isotype, which means that now you have basically the ability to orient in the correct manner those antibodies on the surface. So we solve a technological barrier, and I think from that on, I'm going to show you lots of data, but all the data is basically this technology. And it can go very fast. So basically, if this is a particle, yes, I want to say disregard the ears. Okay, it's a globular particle. What we do in order to avoid some mononuclear phagocytes, some uptaking immune cells, we pegylate those. So this is a pegylated version. In the UK, you can find a lot of those pegylated. And basically now, when I want to orient something, I'm sure this is a good example. When you want to orient something that will be also efficient, this is a great example. If I have two blades like this, it's very hard to cut anything. But if I can orient them, that will be perfect. So this is exactly the type of work that we have done. Behind this were two graduate students. Ronit Kedmi is now in NYU, and Nufar Vaiga in Leuven. We actually manufacture brains outside the country, but hope they will be back. And they came up with this approach, and I'm very quickly going to run, but the idea is now, by switching the antibody, very easily you can go to different cells inside the body. And you can go after specific subsets of immune cells, and we found a way to penetrate them in a very safe manner, which is actually our own advantage. Okay, and behind this approach helped us Itai Benar, who's a protein engineer from Tel Aviv University, and Mark Belke from IDT, who is a great physician, but actually one of the world's greatest nucleic acid chemists. And we started with an antibody, and then destroyed its affinity to create a single chain, and found a bacterial sequence that can lipidate the single chain. So this is basically this unique lipoprotein that can be incorporated into the surface of those particles. Okay, so this is a nice schematic illustration. And now when you have this universal adapter, you can go everywhere. So I'm going to give you some very quick examples. For example, one of them is in colitis. You want to go to inflammatory leukocytes that express specific receptors, we can do it. You want to silence a gene, we can do it. Okay? So this is an example of this IBD model. This is colonoscopy, histology. In any parameter, it actually looks very good. You want to go to mantle cell lymphoma cells? Okay, we can also do it. This is a different receptor, a different target. We can silence this gene, and we have a therapeutic potential. Okay, I'm not going to go into this. Now, we wanted to challenge ourselves and to use this technology in order to deliver mRNA to specific cell types. And I'm going to show you very simply that this approach actually works. This was the first paper showing mRNA delivery to specific cell types. And again, exactly the same idea, the same concept, okay, with this, what we call an asset system. And we can go to different cell type. Again, we chose inflammatory leukocytes, and we wanted to overexpress a protein. This is what you do in mRNA. The idea was, if we can inject this systemically and go to specific cells that are not the target cells, and they penetrate into those cells, now we create those cells, they will become factories to produce this protein. So an anti-inflammatory protein, in this case of IL-10, and basically in all parameters, it also works very well. So in IBD and in other models as well. The other thing that we have done, we said, okay, if we can do this, we can probably also alter the genome. So genome editing is very important, and we experienced just less than half a year ago, Nobel Prize by very talented scientists behind the CRISPR technology. And so we decided to entrap both an mRNA that will be translated into a protein that cuts the DNA with a specific guide RNA. It took us some time, but at the end we found a way to do this and a way to specifically go to specific cell types using exactly the same strategy as I've already showed you. Okay? So we took two models, two animal models. One is a glioma, the other one is a human ovarian cancer. Both of them has different approaches, and this is the work behind this is Daniel Rosenblum and Anna Gutkin. Both left us recently to NYU. And as I mentioned, we have good manufacturing of brain. And this is just an example that one single administration into the tumor bed of a glioma mice that have an aggressive glioma, actually one single injection changed the life span dramatically, and also the editing is very, very high. So it's a very efficient approach. We did it the same with ovarian cancer. And just to show you, here we did 2 injections, and we got dramatic effect in terms of survival. So we can have 2 injections, so that at the end, again, the editing is very high. We can cut the DNA 80%. Just to give you a sense, 6% was reported before of us, and not in a cell-specific manner. So again, we solve a biological problem using engineering approach, and we or I can say also with a biological approach together. Quickly, I think that avoiding mononuclear phagocytes is important unless it's your target cells. High specificity and functionality and fast internalization is very, very important. Lots of things. I'm going to end my talk by showing you a short movie, but before that, the people in the lab that did this job. And so I have to update the slides. And all the funding agencies that help us and some companies that we are collaborating. I'm going to, sorry? I have more, I remember, yes. But these are the ones that were involved in some way in this work that I've just mentioned. And just want to end up with a short illustration movie that hopefully will be more easy to follow than what I just told you.
In inflammatory disorders, the sick cells in our body are our own white blood cells. Only instead of defending the body, they attack it. The best way to fight the complexity of human diseases is to deliver the therapeutic payload directly to the sick cells. A promising technology for combating diseases uses special lipid nanoparticles. These nanoparticles contain RNA molecules that can change the way cells produce proteins and can modulate cells' function. The nanoparticles are supposed to deliver the RNA directly to the sick cells. The problem with today's technology is that the nanoparticles hardly reach immune cells, but rather affect other healthy cells instead. We at Pear Lab have developed a new platform called ASSET that can send RNA nanoparticles selectively to the correct cells. While existing methods of connecting nanoparticles to antibodies are extremely complicated, Pear Lab's method is a matter of simply mixing two solutions. And the nanoparticles are decorated with an ASSET linker, which then binds the antibodies. Each type of antibody knows to attach to a very specific type of cell. So when they decorate the nanoparticle, they will direct it only to the targeted cells. This method means releasing the RNA right where we want it, affecting only the right cells and saving the body from harmful side effects. ASSET nanoparticles are a novel strategy to fight against inflammatory diseases and even cancer. And this time, it's personalized.
Okay, thank you very much.
So thank you, Professor Pear, for your fabulous lecture and presentation. And we will have an intermission of 20 minutes. Please return to the hall by 10:50.